Good morning, everybody. I would like to thank the organizers for inviting me to this participate in this symposium and this conference. And at the same time, I'd like to apologize for not being able to be there in person due to an unforeseen conflict. Brief overview of the topics I want to address in my talk. I'll say a couple of words about some basic facts about the gut microbiome, which in my opinion are essential for understanding the communication with the brain as well. Then address the question of how the gut microbes communicate with the brain, and then go briefly over a few brain gut or a few disorders which have been postulated to be brain gut microbiome disorders. And then finally ask a question, where do we go from here? There's several points about the gut microbiota and the gut microbiome that have become almost common knowledge, not just for scientists, but also in the for the lay population based on extensive coverage in the lay media. Some of these points are shown here. Often underestimated that the inter-individual variation in gut microbial genes is certainly much higher than the inter-individual variation between our human genes. It's also important to remember that the composition, so the question of who is there, that has been addressed in a great majority of studies with the 16S sequencing technology, only gives a limited amount of information about what these microbes do and what signals they can send to the host. On the left side is shown data from in a study from Peter Turnbaugh, the gut microbial composition of identical twins. First two numbers at the bottom and their mother, and there are several of these twin pairs shown here, illustrating the significant inter-individual variability even between identical twins and their mother in terms of the taxa, the dominant taxa, Firmicutes and Bacteroidetes primarily. And on the right side, however, is shown that these very different communities of microbes have a very similar gene pool, basically are able to generate very similar metabolomic signatures. So any estimate on differences and particularly pathological or disease-related differences based on 16S data is incomplete without knowing really what the metabolites are these communities produce. The final point I do want to emphasize is the importance of the early life period in the programming of the gut microbiome and implicitly also on the programming of gut microbiome brain interactions. So on the lower part of this diagram shown the various factors that can affect the development in utero, from infections, medication intake, stress, and nutrition of the mother, the pregnant mother, to effects early on in life in terms of antibiotics and other medications, stress, early adverse life events, to the mode of delivery, and also to the mode of early life nutrition, formula fed versus breast milk fed. The first 3 years of life plus the 9 months of pregnancy, certainly the most plastic period in the development of the gut microbiome and its interactions with the brain, which has significant implications for understanding of many developmental brain-gut microbiome disorders. Here's a summary of the results and the studies that have really started the revolution about brain-gut microbiome interactions. I will not have time to get into the details and the possible limitations of many of these studies. Certainly, they have clearly shown differences in pain-like behavior, emotion behavior, social behavior, and feeding behaviors. And they've also, in some of these studies, at the same time identified associated changes in biological parameters. So not just the behaviors, but also some of the biological changes which have been related to changes in the gut microbiome. One limitation of all of these studies is that they were largely performed in comparing intact microbiome animals with germ-free animals, with many of the limitations of the germ-free animal as a model. So how do the gut microbes communicate with our brain? There's clearly good evolutionary evidence supporting that gut microbes have developed a language billions of years ago when they were the only inhabitants on planet Earth, and that they have used this language in forms of metabolites and neuroactive substances to communicate with the host. About 450 million years ago, first in the digestive tract of the hydra, one of the most primitive animals, and this has turned out to be so successful, the symbiosis, that it has been preserved practically in all life forms ever since. Microbes not only are important for the metabolism of substances that the human digestive tract is unable to absorb or break down, and through this process of fermentation and breakdown of particularly plant-based fiber components, this has been a big positive benefit for the host to extract an additional amount of calories from otherwise lost dietary components. But in addition to this metabolic function, it's now clear that microbial generated substances, neuroactive substances, but also many metabolites with effect on different organs, have an effect on multiple targets within the body, including the brain. And on the right side in this table, you probably can't read it, are shown the various compounds that have neuroactive properties, basically being candidates of signaling molecules that can affect the central nervous system. I'd like to go through a couple of schematics illustrating the components, the two components of brain-gut microbiome interactions in terms of directionality, and then put those together and propose this model of a fully integrated circular process of bidirectional interaction. So let me first show you this concept of bottom-up signaling by cell types and molecules that are generated at the gut level, either directly produced by microbes and entering the systemic circulation, or mediated by the effect of various cells within the gut epithelium, such as enteroendocrine, enterochromaffin cells, immune cells that are activated by the microbes on the luminal side, and then release their contents either into the systemic circulation or onto closely adjacent sensory nerve endings, particularly the vagus nerve. One of those mechanisms I would like to expand a little bit on, the immune cells in the gut. So the largest portion of the body's immune system is localized in the gut-associated immune system, and illustrate here with a dendritic cell that sticks with its tentacles into the gut lumen, has receptors on its luminal side for metabolites generated by gut microbes, such as short-chain fatty acids and secondary bile acids, amongst others. Activation of these dendritic cells then leads to a cascade of immune activation on the basolateral side of the gut epithelium, which then can reach the brain either via the vagus nerve or through the circulation. This mechanism is illustrated here on the top row, that pro-inflammatory and inflammatory substances generated by microbes lead to activation of the gut-associated immune system, ultimately to inflammatory mediators reaching the brain going through or having transduction mechanisms at the blood-brain barrier, activating microglial cells in the brain, which then in turn influence closely adjacent neurons. And this mechanism has been implicated based on animal data, primarily in a variety of chronic brain disorders. Another sort of prototypic cell that mediates this bottom-up signaling is the enterochromaffin cells, shown here on the left, which again have receptors on their luminal side for mediators of the gut microbes, short-chain fatty acids, and secondary bile acids, amongst others, which then lead to the release of serotonin on the basolateral side. The uniqueness of the system is summarized here. Not only is there now growing evidence for a synaptic connection of sensory nerve fibers, primarily vagal afferents, with neuropods, extensions of the enterochromaffin cells that form these synaptic-like structures with these afferent cell types and can send signals to the brain. We also know that signals that come from the brain to the gut, primarily sympathetic nerve fibers, postganglionics, that can stimulate, can activate these cells and lead to the release of serotonin on into the gut lumen. And also through recent really exciting work by Elaine Chow at UCLA, the evidence that certain types of gut microbes, spore-forming Clostridialis included, can stimulate the synthesis of serotonin in these enterochromaffin cells. And based on the mouse experiments, it's been estimated that a significant portion of enterochromaffin cell-based serotonin synthesis is stimulated by these microbial signals. So essentially, this forms a link between the gut microbes, the nutrients that reach the gut microbes, and the nervous system with a direct pathway to the brain, possibly via synaptic connections with vagal afferent nerves. Important, equally important, but often neglected, is the extensive knowledge that we have about the top-down modulation of cells within the gut. So it's been shown that the sympathetic nervous system can lead to release of norepinephrine on the luminal side. And it's also been shown experimentally many years ago that during stress, serotonin in the intestine is released into the gut lumen. So the brain also has this ability, particularly during stressful situations, to regulate the release of luminal contents of these various cell types in the gut. Probably includes the other enteroendocrine cells, and thereby modulate gut microbial behavior and composition. In terms of the serotonin signal, sort of schematically shown here, in response to stress in experimental animals, there's an increase in the norepinephrine, the luminal norepinephrine concentration. We know from many experiments in many pathogens, but also in some commensals, that norepinephrine can activate virulence genes in these in certain microbes, which then lead to an increased interaction of the microbes with the host. Microbes can also, when stimulated, produce adrenergic-like substances that interact with host adrenergic receptors. So again, a full circular communication process between the brain, its stress system, autonomic nervous system, gut microbes, their behavior, and their function, the production of secondary substances. These two communication channels, the bottom up and the top down, are not static in their bandwidth or their ability to transmit signals, but there's two regulatory systems in between the gut, the permeability of the gut, and the blood-brain barrier, both of which are highly plastic and under various conditions can change the amount of information in the gut-brain microbiome This is shown schematically here on the top for an endothelial cell in the brain surrounded by glial cells. The permeability of the endothelial cells has been shown to be regulated by stress, immune activation, and microbial metabolites. On the other hand, at the gut level, on the right side, the schematic showing that there's multiple components to the permeability of the gut wall. One is the mucus layer, or the different components of the mucus layer produced by mucus-producing cells in the gut, but also by the permeability of the gut epithelium itself. On the left side, shown the various forms of epithelial permeability. Most importantly, the transepithelial and the paraepithelial communication channels. I'd like to emphasize that all these components, from the mucus layer to these different types of epithelial permeabilities, can be regulated by the influences shown on the right, including stress, immune activation, and diet, and that there's not one concept of leaky gut, which sort of become a very popular name also in the lay press and in areas such as functional medicine. But these are highly regulated systems, very specifically modulated by these various factors. The take-home message is that what goes on, what's released at the gut level and what gets into the brain and vice versa is highly plastic and influenced by the context of the Putting everything together, we have a model that shows a fully integrated brain-gut microbiome-brain communication. Any event that happens at the brain level in terms of emotions or stress will have a reflection at the gut level, and any perturbation at the gut level will, through afferent multiple communication channels back to the brain, alter brain circuits, if it's chronic brain structure as well. This is important because I think we still adhere to a linear concept that if something happens at the gut level, there's sort of a simple ascending loop without taking into consideration it's always associated with a response to the brain that feeds back to the gut, and it's the gut connectome. Primarily based on animal models, there has been several brain disorders have been identified as putative brain-gut microbiome disorders, which includes irritable bowel syndrome, but includes also many common psychiatric and neurological diseases. I do want to emphasize that much of this evidence is currently based on animal models, on association studies, but there's a slowly growing number of human studies that suggest that there's clearly a correlate at the human disease level. So I'd like to say a few words about disorders of mood and affect, particularly depression. Early studies had shown that behavioral traits can be transferred from one strain of mouse that has a particular emotional behavior to germ-free mice that do not have that trait, which then results in a development of behavior in the recipient identical to the donor animal. There's now recently have been 3 studies in psychiatry in the area of depression, clearly have identified differences between patients with major depressive disorders and healthy human subjects in their gut microbial composition, illustrated here with a surprising clear separation between the healthy subjects and the patients. In this study by Sheng, it's also been demonstrated that if fecal material is transferred from the patients into germ-free animals, that there's also a similar dysbiotic state can be induced in the animals, shown here. And this is also reflected in by metagenomic analysis and also in a identification of the proteins, the enzymes that stool from depressed patients can induce significant changes in the mouse recipient. And that was associated with animal behaviors that are just taken as evidence for anxiety, increased anxiety-like and depression-like behaviors. Findings similar to the Zheng study have now been reported by two other groups, one from China, one from Ireland. So I would say that in the human domain, the evidence that the gut microbiome plays some role in human depressive disorders is probably best established for this particular syndrome. One model that one could explain these findings is that altered influences from the brain, with brain changes being the primary abnormality, can induce changes at the gut level, the gut connectome with multiple cells adapting to this altered influence, which could be increased sympathetic input chronically from early life on, that the output then of the gut connectome generates metabolites that feed back to the brain and reinforce the central abnormalities. Once established, this abnormality clearly then is at the brain-gut microbiome level, and interventions on either side of the brain-gut axis could potentially be beneficial, including combination therapies, for example, in terms of nutrition or novel probiotics and behavioral or pharmacological therapies at the brain level. Parkinson's disease, another disorder where there's growing evidence for an involvement, for some involvement of the gut microbiome. Shown here, the time course of Parkinson's disease, which is now well known to have involvement of non-brain parts of the body early on, often decades before the onset of typical clinical symptoms of Parkinson's disease. And the gut is one area where such abnormalities can be identified clinically presenting as depression. Addressing the question of how a signal that starts, an abnormal signal that starts at the gut level could make it to the brain. Evidence, these epidemiological studies showing that a truncal vagotomy in patients appears to be protective against the development of Parkinson's disease is shown here, whereas a highly selective vagotomy does not involve the intestine, did not have the beneficial effect. And models have been proposed that the so-called Lewy bodies that are initially detected can be detected in biopsies of patients with very early forms of Parkinson's disease. Ultimately, the signal makes it to the brain, and that the vagus nerve is one pathway, one channel that this signal is transmitted. What the mediators are, if these are viruses, prions, or other factors, is currently not known. Should also mention that a recent study by the Masmanian lab has identified short-chain fatty acids as a possible mediator in the development of central changes in Parkinson's disease. There's also some evidence from our own studies in irritable bowel syndrome, evidence that and still the only evidence that we generated a couple of years ago, that perturbation of the gut microbiome can change brain function. This was done by the ingestion, clinical trial involving the ingestion of a probiotic mix for 4 weeks in healthy young women, led to a change in the emotional reactivity at the brain level, identified as a decrease in the connectivity of a brain network engaged in response to an emotional attention task. Should mention that this was not associated with a change in the gut microbiome, but previous studies have identified changes in the metabolites produced by the gut microbiome in response to a 4-week ingestion of this particular probiotic. We've also found associations, and this is has clearly the limitation of being an association studies, of gut microbial compositions identified in a subgroup of IBS patients that had a dysbiotic state, as opposed to a second subgroup that had an identical gut microbial compositions to healthy controls. That there was an association of gut microbial composition with brain structure for the structure shown in these images. So if this is something that develops early on in life, when both the gut microbiome and brain circuits are being formed, or this is something that could develop later under chronic conditions of abdominal pain and discomfort, will need to be established. The last piece of evidence that I would like to mention here is the role of gut microbes in obesity and ingestive behavior. There's again multiple pathways that have been identified between the gut and the brain in terms of satiety signals, which are in turn modulated by signaling molecules such as short-chain fatty acids produced by microbes. It goes back to early animal experiments where it's shown that a genetic model of mouse that is obese and also shows uninhibited feeding behavior could be transferred to a germ-free mouse that was non-obese, leading in the recipient to uninhibited eating and the development of obesity, suggesting clearly that this is driven primarily from the brain and not from an increased harvest of undigestible food. So in these studies done by Claudia San Miguel and our group, it was in patients undergoing bariatric surgery, so patients with morbid obesity, shown that the surgery not only led to pretty dramatic behavioral and changes in food preferences, shown here, even within 1 month after surgery, that these changes were associated with pretty dramatic changes at the brain level, showing the connectivity map of the brain before and after surgery, showed a much significantly decreased connectivity pattern between the basal ganglia, the nucleus accumbens, and other brain regions, probably the mechanism underlying the change in behavior and the change in food cravings. A change in tryptophan metabolites in the gut level, and a correlation of these metabolites that were altered in the postoperative with both the brain changes and with behavioral changes. So in my opinion, this is one of the best evidence now in humans that substances, signaling molecules produced by the gut microbes have an effect on the brain, resulting in pretty dramatic behavioral changes, and clearly with major implications, potential therapeutic implications, not in form of surgery, but in targeting some of these involvements. So as opposed to depression, and I'm almost at the end here, one could postulate that in the development of abnormal eating behaviors, particularly food addiction leading to obesity, that the system really starts, the problem starts at the gut level with with ingestion of a diet high in animal fats and sugar, which leads to initially to a change in the gut connectome, which then leads to altered signaling patterns back to the brain, which then changes brain circuits towards a predominance or increased functional connectivity of reward circuits in the brain, which will continue the vicious cycle of leading to increased ingestion of the unhealthy food. So depression and food addiction, two models, potentially one starting at the brain level, the other one starting at the gut level, but both involving alterations in gut microbiome, in brain-gut microbiome. So there's several questions that, several areas that are likely, are being addressed now by multiple investigators. So clearly one has to do with, in longitudinal studies, to look at causality. First of all, to reproduce some of the findings. Secondly, to look at causality. Do these disorders start at the brain or at the gut level? Also, identification of distinct metabolomic disease signatures, and the development of novel therapies based on clearly identified targets and mechanisms. How far we are away from this goal remains to be determined. Certainly, there has been a lot fewer human studies that have supported some of the animal studies, but in the last couple of years, clearly now there is growing evidence that in a few of the human disorders that have been postulated as brain-gut microbiome disorders, there is also the human evidence. So I'd like to mention some of the collaborators that have contributed to the results I've presented to you. And finally, if you want to know more about these multiple connections between the brain, the gut, and the microbiome, take a look at my book, The Mind-Gut Connection, which summarizes many of those and expands on many of the topics that I mentioned today. Thank you very much.